Hey, what's up, biochemistry classmates? So in this video, we'll be talking about amino acid catabolism 2, otherwise known as the urea cycle. So if you guys recall from the first video, one of the major threats or major problems that we face whenever we use amino acids as an energy source is that amino group, because in our amino acids, the only way it can be useful is if we get rid or we strip that amino group off, that nitrogenous product off, right? However, this is bad because if we just strip it off and let it get into our bloodstream, ammonium ion is actually toxic to us. And we'll talk about why in this video. And so we are going to talk about the urea cycle, which is basically a way for us to allow for amino acid catabolism, but also safely excrete these nitrogenous products in a safe form known as urea. And we basically just pee it out, right? It goes to our kidneys and then we pee it out. So the first three objectives for this set is basically know the urea cycle. There's not really a shortcut around it, but yeah, know the urea cycle, know all the enzymes used, the coenzymes used, know your substrates, know your products, know the location of where everything happens, and yeah, and it's kind of complicated. It's interconnected with the citric acid cycle as well, so it's confusing in that way, but it's not impossible, I don't think. So the urea cycle. Let's just get started. Hopefully this doesn't take too long. Essentially, we need to get rid of our nitrogenous products, right, in the form of urea, right? And the way we do that is we use aspartate, which has this green nitrogen right here. And you'll see we'll follow this green nitrogen all throughout, and you can see where it ends up in urea, right? And carbamoyl phosphate, carbamoyl P, carbamoyl phosphate, which is the blue nitrogen, which will follow all throughout here. And as you can see, it ends up over here in urea as well. So before we even talk about the urea cycle, let's talk about how we make the substrates that are needed to excrete urea, to make urea to excrete it, okay? The two most important substrates in the urea cycle are your carbamoyl phosphate and your aspartate, okay? And this is because these are gonna be the sources of nitrogen we use to make urea. So here's the blue nitrogen of carbamoyl phosphate all the way over here, and here is the green one down here, the green one that started all the way up here at aspartate. And so how do we make aspartate, or how do we make carbamoyl phosphate? This is all up to glutamate, okay? So glutamate can actually be used to make aspartate or carbamoyl phosphate, all right? And so that's why glutamate is such a key component, a key amino acid in this urea cycle, and such a key component in how we can use amino acids as a source of energy, all right? And so let's just start from the very top here, right? So we're going to use some amino acids, right? And we're going to basically do this transaminase process, which we talked about in the last video, and we're going to create a keto acid with our amino acid. But then we also create a glutamate, right? So our alpha ketoglutamate will turn into a glutamate group, right? And this is the same thing with alanine, okay? Except alanine will turn into an alpha keto acid, specifically pyruvate, which can also enter the citric acid cycle. But at the same time, we're also transferring that amino group onto our alpha ketoglutarate to make glutamate, all right? So whenever we have amino acids coming from like our diet, or alanine from breakdown of our muscle, we're going to make glutamate, and glutamate will be shuttled into the mitochondria to eventually make this carbamoyl phosphate, remember what I talked about, and this aspartate, the two important molecules that will supply the nitrogen to our urea cycle, okay? So the other way that we can get glutamate is glutamine. So in other tissue other than muscle, and sometimes muscle, glutamine will come in, and glutamine will turn into glutamate in the mitochondria, and we use the enzyme glutaminase. Glutaminase? I don't know, glutaminase, but essentially this first step, we lose a nitrogen ammonium ion, and then we make glutamate, all right? And this ammonium ion can actually be used to make carbamoyl phosphate once again, all right? Okay, so now we have transferred our nitrogenous product onto glutamate. Now what happens? So now glutamate has two fates, right? It can either become carbamoyl phosphate or it can become aspartate. It can apply nitrogen for carbamoyl phosphate, or it can apply or supply nitrogen to aspartate. So let's say it applies it to aspartate. What we're going to need is oxaloacetate first, all right? So glutamate and oxaloacetate will combine, and we're going to use the enzyme aspartate aminotransferase, and we're going to make aspartate this way, okay? All right, so aspartate has this green nitrogen that we'll see later in urea, all right? The other way is it can become alpha-ketoglutarate, right? But it can supply another nitrogen, which is needed in this reaction right here, this CPS1, all right? So CPS1 stands for carbamoyl phosphate synthetase 1. I didn't have room to write it in here, but that's carbamoyl phosphate synthetase 1. And you're essentially going to use 2 ATP, an ammonium ion that either came from your glutamate or came from your glutamine, and bicarbonate. And you're going to use this enzyme and you're going to make carbamoyl phosphate, okay? So now we have all the ingredients, at least in terms of nitrogen, to go through the urea cycle. So this is amino acid catabolism. Now let's talk about urea cycle. So now we need to make citrulline to start. But before we can make citrulline, we need ornithine. And where does ornithine come from? It comes from our amino acid arginine, okay? So arginine is gonna have acted upon it arginase, all right? So notice the color schemes here. Purple will be the urea cycle. These black arrows up here, these were the amino acid catabolism steps before urea cycle. And then notice the blue nitrogen, follow it throughout this cycle, and the green, all right? And then these in blue, right here and right here, these are our enzymes, okay? In the lightish blue, all right? Okay, so now we have ornithine. We're going to combine ornithine with our carbamoyl phosphate, and we're going to make citrulline, okay? And the enzyme that's responsible for this is ornithine transcarbamylase, okay? All right, so notice in our citrulline right here, it's just going to be a combination of our ornithine and our carbamoyl phosphate up here, but we lost the phosphate group, okay? So our blue nitrogen is now on our citrulline. Citrulline, this is done in the mitochondria, but then citrulline will be transported out into the cytosol, where it's going to undergo the reaction with arginosuccinate synthetase, all right? Now we use our other ingredients. So the two ingredients, remember, were carbamoyl phosphate. The second will be aspartate. Aspartate will also be shuttled out of the mitochondria, and now it can be used to make arginosuccinate. So this is our aspartate up here with red and green. You can see how it's attached in red and green down here to our citrulline molecule, okay? So red, green, blue, and black, everything is there now. All we have to do is get rid of those nitrogenous groups, okay? So the next step, we're going to use this enzyme argininosuccinase, and we're gonna poop out this red group right here, and that's gonna make fumarate, okay? We'll come back to this later, but what we're left with is arginine, all right? So we're back at where we started earlier, where we use arginine to make ornithine, and ornithine to make citrulline. And then this up here is we're getting supplied with aspartate and carbamoyl phosphate by amino acid catabolism. Okay, so final step, right? Arginine back to ornithine. Using arginase, we have to add water, and out pops the urea group. So now we successfully have made a urea molecule with two nitrogenous substances, and now it will travel to the kidney and will pee it out and excrete it safely, all right, as waste product. So this is the urea cycle. It's very long. This picture is in your textbook. I would literally draw it, and then when test date comes around, I would try to recall everything from memory. It sounds really hard, but this is what I've been doing for all my pathways in biochem, and usually around the third try, I have almost everything down. I think, yeah, just give it a try. I literally go to Dane Smith sometimes, and I'll use the chalkboards and just recall it from memory. And this is, I think, in my opinion, the best way to learn it or memorize it. And literally memorize everything: the structure, how to draw aspartate, how to draw citrulline. Yeah, and know in each step what is happening, right? So you're adding this to this, or you're adding that to that, or you're taking this out, you're oxidizing this, you're reducing this, and that'll help a lot, I think. So that's the first three objectives, guys. All right, guys, so moving on to the next objective, objective 4, draw/describe how the citric acid cycle and urea cycle are integrated. So this is a nice little picture for us here, right? But if we actually go back to our picture, right, we see here that alpha ketoglutarate made in amino acid catabolism that can enter the citric acid cycle, right? Fumarate, which we made out here, can enter the citric acid cycle as well, right? And actually, fumarate can enter the citric acid cycle and be turned into oxaloacetate, and then using aspartate transaminase, we can make aspartate again. So from the citric acid cycle, we can make aspartate, and remember, aspartate is one of the important precursors or supplies of nitrogen in our urea cycle. So just from this picture alone, we can see where things are sort of connected, but this picture kind of shows it more compact. Okay, and so here, yeah, so oxaloacetate, right, can turn into aspartate, which can be transported out of the mitochondria and enter the urea cycle, right? Or, right, arginosuccinate makes fumarate, right? Fumarate can be turned into malate, which can be transported using the malate alpha ketoglutarate transport system and enter the citric acid cycle. So these are really the only two major integration parts, but yeah, so this is a nice little illustration of that. Last objective for this set: what is the point of the urea cycle? Why is it worth expenditure of ATP, and how much ATP is needed? So essentially, in our brains, we have these cells called astrocytes, right? And in red here, I've sort of expanded or blown up this astrocyte cell for us. And in these astrocytes, we have an enzyme called glutamine synthetase. And this glutamine synthetase can turn glutamate into glutamine. And it uses your ammonium ion, which is excreted right here in amino acid catabolism, right? Right here, right here, right? And it makes glutamine. And essentially, glutamine is osmotically active, okay? So the more ammonium ion we have in our bloodstream, the more that's sent to our brain, the more glutamine we're going to make. And if you remember from Gen Chem with osmosis, if you have a semi-permeable bag and you put salt in it, so the salt can't pass through but water can pass into it, this bag is going to expand. So what's going to happen to our brain here? It's going to expand inside our skull, and that's not good. This is known as cerebral edema, and it can lead to intracranial pressure, which is bad, can lead to things like stroke, or like sometimes even comas, or maybe in the worst case, death, right? And so this is very bad. We do not want this. Urea cycle is very important if we're using protein as a source of energy. And actually, this is one thing that bodybuilders need to look out for. If they're taking in too much protein and they're metabolizing too much protein as energy, this can be one of the outcomes of it. So how much ATP is actually needed? 4 ATP is actually needed to run the urea cycle. So the first 2 is right here, right, in this carbamoyl phosphate synthetase step. And the second 2 come from the citrulline step, citrulline to arginosuccinate. You use 1 ATP, but you actually use 2 phosphate groups to make an AMP. And so this is essentially like using 2 ATP. So in total, we use 4 ATP. Is it worth it? Yeah, because ammonium ion is very dangerous, right? All right, so that's all we have to know for urea cycle. There is a section on regulation of it. I would read it. It's pretty short. It wasn't in our objectives, but I feel like it's important. So yeah,